So please, Daniel.
Thank you. Thank you. I appreciate Bent setting up this discussion. I can skip through a lot of my first slides. This was designed to be a standalone presentation. It will be made available, so you'll have copies to use as you see fit. First question may be, what does curated mean? There's a gatekeeper. Only studies that have been reviewed for reliability are in the database. We intentionally set it up that way. Use this, okay. What I want to do today is just very briefly talk about why we developed the database, a little bit of information on the Water and Environmental Technology Center that houses this database, and some of the benefits and uses of the database. You've seen this from Bent. Only 2 points I want to make. I think that the Sum of Us report is the best report I've seen in probably the last 10 years. It's well written. The graphics are impactful. We need to see a lot more of this kind of information to help us get our message out. And as Bent mentioned, SICOM had a decision in October of last year to adopt environmentally persistent pharmaceutical pollutants as an emerging policy issue. And then the week later, there was an editorial that appeared in Nature. The bold text is mine. You can read that at your leisure. But it kind of sets the stage for what we are up against as an industry. A lot of perception that we do nothing, we have no information. As I hope to convince you shortly, that's not the case. I like this graphic also because in addition to the 4 different reports, you can see that there's a clear focus on manufacturing on a global basis now. It's not just what we make in our own backyards, but it's what we outsource from external suppliers. To address that, we've got the eco-pharmaco stewardship, particularly the effluent management pillar. But to establish risk-based controls and share best practices, we had to do a couple of things. So we published 2 papers recently. One came out in April, the second one's coming out in May, Environmental Toxicology and Chemistry, where we published a wastewater maturity ladder and a method to evaluate manufacturing discharges. And as you look through some of these, and if you get to step 6, when you look at toxicity identification and doing the assessment, you have to have a basis. You have to have a predicted no effect concentration. So that's why we developed the curated database. The paper by Jessica Vestal will be coming out next month, May, Environmental Toxicology and Chemistry. The database is based on that paper, plus additional data that was added after this project started, which has been about 5 years now. So what we have now are acute toxicity data, the standard data set you have to develop for any chemical. It's used for classification, transportation. More importantly, it's the chronic information. That's what you really need to set a good predicted no effect concentration. The sources predominantly were unpublished company studies that were done by OECD or US EPA guideline or FDA guideline in some cases. The same sorts of studies we have to do to get a new drug approved. Some of the data is publicly available. AstraZeneca has their information on their website. We chose to put our information on the university website because it makes it a lot easier to maintain the ability to have public accessibility. We used the data from FAS, and then for the paper purposes, where we had data gaps, if we didn't have 3 species, we went to the literature, did a review, and then there was a lot of right-censored data, a lot of no effect concentrations that are greater than, and that posed a lot of statistical problems. So we ended up consulting with a statistician and came up with some solutions. So in the database itself, this is what's there. We broke it down by the American Hospital Formulary System for drug classification. We initially tried to do a lot of work with acute to chronic ratios. In some cases they work, they're different by species. So we pretty much abandoned that and went strictly then into mode of action of various compounds and what the data show. And this is the take-home message. From the data that we have, you can see that everything below that line on the bottom, which is a predicted environmental concentration on the EU basis, 10 nanograms per liter, the compounds that you would expect to be problems tend to show up with very low PNECs. Estrogens, anti-estrogens, and a lot of anti-neoplastics that have an endocrine mode of action. Again, these numbers are based on data. What good is this database? Well, you know, to me, it reinforces the value and the utility of having good acute data. If you derive a PNEC on the acute data with an assessment factor of 1,000, in most cases, with the exceptions obviously of the endocrine active compounds, you're going to have a more conservative PNEC than you are if you do the chronic studies and put a factor of 10. You know, moreover, if you use these data in an assessment, it will help you target where you need to come up with additional information. We think that in general, the pharmaceutical data set requirements for environmental risk assessment are fit for purpose. What's in the Medicines Agency guidelines is a good place to start. Obviously, as you can see from the previous slide, if you've got an endocrine mode of action, you probably want to do additional work, most likely in fish. So moving on to the Temple University Water and Environmental Technology Center. This is an industry university cooperative research center which is funded by the U.S. National Science Foundation, member companies, and also some of the universities. This particular center has 3 universities, Temple in Philadelphia, Arizona State, University of Arizona. They have different aspects, different focuses on water. Among the industrial members, Johnson & Johnson and Pfizer, and there are also several sanitation districts, wastewater treatment, municipal wastewater treatment systems, as they're called, that are members of this group. The website is do you have internet access on a computer? I can show you the database if we have time. Or we can do it during a break. But you can go to that site. And you can get a background on the facility. You know, again, the benefits and uses of the database, increased transparency. Yes, there are data for a lot of pharmaceuticals. They just haven't been publicly available before. I hope to have it up. It's probably going to be June now, a list of PNECs that you'll be able to search, publicly accessible. And then for the underlying data, we will make that available to environment agencies upon request. The types of studies and the NOACs from the individual studies. We think that this information will be very useful to looking at local environmental circumstances. We're using this as part of our capacity building efforts within the Pharmaceutical Supply Chain Initiative. When we talk to our suppliers in India, in China, other places that don't have the same level of environmental standards that we have in the rest of the world, you can use this as a screening tool. You can also make this information available to local regulatory agencies. You know, and if the Indian government wants to set a predicted no effect concentration or an environmental quality standard, they'll have access to the data to do that. And then lastly, we think this is a good way to prioritize additional environmental risk assessments. If you in a member state or another country have monitoring data and you want to see if that could be a risk or not, you can have access to the information so you can do your PEC, or in this case, measured environmental concentration, to the predicted no effect concentration and do your risk assessment. Lastly, the citations for the 2 papers, and I'm happy to answer any questions.
Thank you very much, Daniel.
You're welcome.
I have one clarification I want to start with. The PNEC that you are working with, that will be very interesting, and I'm very curious to get the feedback then from people who are more competent than myself to evaluate and digest that data. But is that still toxicity based? and substance by substance, because that's the key critique that we hear against the whole concept of the PNEC. It's toxicity based and it's single substances. So what's your take on biological or pharmaceutical effects and the so-called cocktail effect? Is that handleable there?
There are certain classifications, certain categories of pharmaceuticals that would have additive effects. The estrogens, obviously, they have the same mode of action. Narcotics, narcosis is a pretty basic mode of action. Things that act by the same method could have additive effects. One of the things we're working on is a biomonitoring project with, again, colleagues at Pfizer, Teva, the Swiss, the Italians, to look at a bioassay to monitor surface water. Rather than going out and trying to measure very low levels of individual chemicals, if you can do a bioassay and see some effect, then you can focus efforts to go and measure, because you're talking for picograms per liter for ethanol estradiol as being the EQS proposal in Europe right now. So before you go out, collect a lot of water and show that you've got below limits of detection, it would be good to be able to look biologically to see if there is an effect.
Mm-hmm. Thank you. Joachim, and any further questions?
So thanks, Dan. I was just wondering that you suggest here publicly accessible list of PNECs, but the underlying data and the NOACs and the study type is not suggested to be publicly available for university researchers worldwide, etc., but for specific regulatory agencies. And I wonder, why is that? Why is there a sort of a limited access for the rest of society that is not within a regulatory agency?
It's a work in progress. We, I'm sure, can make data for compounds of interest available for researchers. I also have an ulterior motive, and that is I want to get more pharmaceutical companies to join. So rather than give away the data, you can have the PNEC. You want the data? Join. What I thought you were going to ask is, what about antimicrobial resistance? And I think that's a incremental improvement moving forward once we get a firm basis. Several of my colleagues at different companies have different methods to do just that. So we're in the process of trying to identify what's the best way, and I'm well aware of your paper. And that's one potential addition. The other one is potentially looking at a drinking water safe exposure limit. That one may be a tougher sell, but again, that's an option to add that into that.
Okay. One more question that I usually keep irritating Bengt with. Why don't you talk about zero liquid discharge instead of putting quite some effort in estimating the best or highest acceptable level in the effluent? Wouldn't it be much easier to say just we don't release anything, point?
Well, the areas of the world that have a zero discharge end up having other issues because the water has to go somewhere. And when you inject that into a water-starved area and the water is drawn up, it's better to do an assessment on a risk basis. I mean, you're never going to get to zero for anything. That's why we focus on what the risk is. And that's why we maintain the database as being a curated database, so we're comfortable with the data that's in there. And again, tying back into the enhanced environmental risk assessment, if additional data become available later on, and a good example of that is ethanol estradiol. I saw the original 1997 environmental risk assessment. It's one 3-sentence paragraph. It said, based on the current levels of exposure, we don't anticipate any problems. Well, you know, hindsight's 20/20. Knowing that it functions by binding to estrogen receptors, and knowing that there are estrogen receptors in all fish species and mammalians, you could have perhaps had a little more proactive approach in potentially identifying additional targets to look at. So here we are, what, 20 years later, we have a lot of data on ethanol estradiol.
Thank you very much.
You're welcome.